Good afternoon to everybody. Today I am going to discuss about the topic that is plant regeneration pathway. Plant regeneration pathway through process of organogenesis and somatic embryogenesis. It is a part of plant tissue culture. So, what are different methods for regeneration, for multiplication of the plant? First is organogenesis and second is somatic embryogenesis. So try to know the different organs that develop through plant tissue culture. This is the callus. Okay, you know, this one is the multiple shoot formation from the explant through plant tissue culture. Okay, and this one is the somatic embryo, and this is the protoplast, cell without cell wall. Okay, and this is the somatic embryo that have different stages when they are growing, and due to course of time they are changing their shapes. Globular stage, that is gada shape, and this is the heart shape, that is the shape of somatic embryo, and then this one is the torpedo stage. So this is the different sequential steps, one by one they are attaining from the somatic embryo. Initially it was a callus, now this callus developed into somatic embryo. uh, the callus developed from explant developed into callus, and callus developed into somatic embryo. And this somatic embryo, they change in the shape. That is, what is the shape? That is the globular stage, that is gada shape. And then further into the heart shape, just like you will find the heart shape like a structure, and you will find also the torpedo stage, that is the sequential step for the growth and development of plants from somatic embryogenesis, whether we are utilizing only somatic cell, not reproductive cells. So these are the method through which we can rapidly multiply the plants. What are different method? One is somatic embryogenesis, second is organogenesis. So try to know, you know, these are the apical portion that is meristematic in nature, that has capability to divide. So generally we choose the portion of the plant that have ability to divide and multiply, that is known as apical meristem. And we can use this meristem, apex, the nodes and other explant, and we multiply this plant part organs, that is known as organogenesis, means development of the particular organ from explant, that is known as organogenesis. And development of whole plant from the explant, that is known as organogenesis. So chemical nutrient play a vital roles, means media, and that media contain the growth hormones. The main factor growth hormone is auxin and cytokine. If there is equal concentration of auxin and cytokinin in the media, then you will find the structure like that is callus. This one is callus. They undifferentiated. We cannot differentiate it whether it is the root part or a stem cells. So that have ability to divide and multiply, but we cannot demarcate it is a stem cell or root cell. It is callus. Then further we decrease the concentration of cytokinin or decrease the concentration of auxin, anyone. If the high concentration of cytokinin, it favors the stem formation, it favors the stem formation, that is shoot formation from the callus. Initially it is explant, and then it form the callus, then we develop the different organs. And then if there is high concentration of auxin, then it form the roots. So rooting formation occurs. So you will find the somatic embryo have the different shape, that is the globular stage, that is the torpedo shape or heart shape, somatic embryo. And finally you will get the cotyledon, and from that you will get the whole plants. So these are the different method for regeneration of the plant. One is organogenesis and one is somatic embryogenesis. You will find the plant part, and you can take any part for regeneration of particular organs or whole plants. So these are the flowchart that depicts the formation of particular organs. You will find here in this diagram. And then from the callus, first there is development of callus, and from the callus it is going to form the either stem or the callus or the root formation. It will depend upon the concentration of cytokinin and auxin. If there is high concentration of cytokinin, it will favor shoot organogenesis, and if there is a high concentration of auxin, then root organogenesis. Means we are going to form particular organ through organogenesis. From so it was first of all reported, it was first of all reported by the scientists Spook and Millers. Okay, so first in 1944, 1944, the F. Spook had identified the in vitro organogenesis regulated by chemicals, and the Millers in 1957 also identified the same thing. The organogenesis means formation of particular organs from the explant in the presence of particular chemical, that is growth regulator. It can be auxin or cytokinin. If we utilize the high concentration of cytokinin, it favor the formation of shoot. If high concentration of auxin, it will favor the formation of root. So you will find these things means the scientist who was associated with the formation of organogenesis first time, it was given by uh, F. Spook in 1944, and then further the Millers in 1957. In high concentration of auxin and cytokinin favor the root formation, and high concentration of cytokinin and auxin that favor the shoot formation. He has done this experiment, they have done this experiment on the carrot. Next, somatic embryogenesis. You see this slide. So this is a clear cut demarcation, means how there is a formation of the shoot or root. Okay, so if you are facing any problem, then you can ask questions. If you are observing any difficulty, then you can ask. So try to know the um, just see, this is the stage of somatic embryogenesis. What is somatic embryogenesis? We are going to form the whole plant part from the explant. Explant means it can be a leaf, apical leaf part, or apical stem part, or apical root part, but we are going to regenerate yes, so we are going to regenerate the whole plant part from the
नहीं देख लो।
explant. सुनाई नहीं पड़ रहा है क्या? आर यू लिसनिंग माय
सर
साउंड?
आपकी आवाज कट कट के आ रही है सर।
अच्छा अच्छा ठीक है, तो सारे लोग ऑफ कर दो, फिर आवाज आएगी. अभी ये स्क्रीन दिख रही है ना, स्लाइड दिख रही है, स्लाइड दिख रही है ना?
साइड भी नहीं दिख रहा सर।
Yeah. So this slide, that flow chart indicate the formation of embryogenesis. So that is somatic embryo, not embryogenesis. That is somatic embryogenesis, means we are going to produce a whole plant part from the explant. So what happens? First, explant we put in the suitable media that contain equal concentration of auxin and cytokine, and then it will develop into callus. So that callus we use, again we put in the media, and there is a formation of different stage of embryo, somatic embryo, that is the uh, globular stage, that is the gada shape, and then heart shape stage. You field, the stage, the shape of the embryo, and then finally this one is the torpedo stage. So that is going to develop into cotyledons. So cotyledonary stage, and from this there is a formation of stem or shoot in condition of high concentration of cytokinin. Try to listen the word. Cytokinin is the hormones that proliferate for the formation of stem part. If there is a high concentration of cytokinin, then there is a proliferation of the stem part. But if we are changing the media that contain high concentration of oxygen, then again there is a formation of root. So we get the whole part of the plants, the stem part, shoot part or stem part, and root part. So we get the whole plant through artificial medium from somatic cell, means the leaf cell or the stem cell or the root cell. Not we are going to use the fertile seed, means we are not going to utilize the seed portion for multiplication of the plant, for regeneration of the plant. Multiplication or regeneration both are same, means you are going to maintain the plant population of any plant species. It can be a rare species, it can be an endangered species, means we are using the plant tissue culture methods. Okay, next try to know in this lecture factor affecting organogenesis, advantage of organogenesis, application of organogenesis, somatic hybridization. So try to know the term organogenesis means development of particular organs, either root or as a organ or shoot as a organs or leaf as a organ from the somatic cell, that is known as organogenesis. It was first of all reported by scientists F. Esco and C.O. Miller. So in 1957, C.O. Miller observed the formation of a stem in presence of cytokinin. And then what is somatic embryogenesis? Formation of the whole plant part from the somatic embryo. What is somatic embryo? Somatic embryo, it is produced from the explant, newly growing cell, that is meristematic cell, first converted into callus, and then gradually callus develop, multiply, and form the somatic embryo. And from that somatic embryo we are going to multiply the plants. Okay, so this is the flow chart of the somatic embryogenesis, means we are going to utilize the somatic cell, not reproductive cell. So try to know what is organogenesis? Process of differentiation by which plant, root, shoot, bud, flower, stem are formed, while advantageous refer to development of organ or embryo from the unusual point of origin of an organized explant, where a preformed meristem is lacking. Means we are going to multiply the plant from the explant that have capability to multiply and divide. Means we are utilizing the somatic cell, not reproductive cell, to develop a root portion, a shoot portion, a bud, flower bud, or a stem portion as per the changing the hormonal concentration. What hormones? Oxygen and cytokinin. If you utilizing, if you will utilize high concentration of cytokinin, stem formation, high concentration of oxygen, root formation. This process is much more common than somatic and has for more potential for mass clonal propagation of the plant. Means it is common, and we can easily multiply the plant with the help of somatic embryogenesis than the normal embryogenesis. Normal embryogenesis means transfer of pollen grain to a stigma of the flower, then there is a fertilization, then zygote formation, then that zygote develop into embryo. So that is known as zygotic embryogenesis. Common than here it is wrongly written, it will be the zygotic embryogenesis. So zygotic it is a rare event, we can not easily multiply. It will depend upon the season, on the season only, particular season, in particular season only we can multiply that plant through normal conventional method. But we can utilize somatic embryogenesis in any season for multiplication of any plant with the help of the hormones, plant hormones. So what are the factors that affecting the organogenesis? There are so many factors: physical factor and chemical factor. So try to know what are the physical factors: the intensity of light, then temperature, then electric stimulation. Means we are utilizing the different temperature for different variety of plants as per the uh, uh, reports, research. So many research is going on regarding the plant tissue culture. As per the variety and of the plant, we select the temperature and then intensity of light, whether 16 hours intensity of light is required or not. Then we can also use the phytochrome, that is electric, means that will take the they take the light for formation of flower bud, that phytochrome, for flowering, for anthesis, that play a vital role. Next is chemical factor. You will know, I have already discussed these things. This cytokine and oxygen are plant growth regulator, or plant hormones that play a vital role in organogenesis and somatic embryogenesis, that lead to formation of bud differentiation. Means cytokinin mainly responsible for bud differentiation. Then so kinetin, that has 30,000 times more potential than adenine for stem formation or bud differentiation. And you will find the phosphate increase. The level of phosphate in the media is reported to conduct the inhibitory effect of oxygen. Means if you are going to increase the oxygen, then there is a root formation. But if you are adding the phosphate in the media, then it will inhibit the action of oxygen, that is responsible for root developments. Casein hydrolysis or tyrosine also induce the kinetin type bud formation even in presence of high level of indole acetic acid. Indole acetic acid, that is the oxygen. So that is helpful for the bud formation. So generally these are the chemicals that play a vital role, act as a hormones. But you know the mainly two terms, that is oxygen for root formation and cytokinin for shoot formation. If there is equal amount of oxygen cytokinin, callus development. If there is a high concentration of cytokinin, shoot development. And if there is a low concentration, high concentration of oxygen, then root development. So cytokinin for shoot, high concentration, and oxygen for root formation, high concentration, but equal amount it will responsible for callus development. So these are the organogenesis. You know, shoot formation, root formation mean development of particular organ as per the changing the media. If you will not change the hormone concentration, there is no demarcation whether it is the root or a stem. It will remain as callus. So you, in this case, from the callus, when you put the callus in the media containing high concentration of cytokinin, then it will develop as root, shoot. And if we putting the callus, because callus we are getting from the explant, and we put the media in the container that contain high concentration of oxygen, then there is a development of shoot formation, division of shoot tissue, and that develop into shoot system. And then that we develop the whole plant. First we develop the shoot portion, then root portion, then rooting, shooting again, and then the whole plant develop. So try to know cytokinin for shoot formation, oxygen for root formation. But equal amount of oxygen cytokinin, neither shoot formation not root formation, only callus. So callus is the initial, just initial stage that develop from we develop from explant, and explant is meristematic. So we get the whole plant after development of shoot. First we develop the shoot and then develop the root. Next, you see the stage of organogenesis. So just see in the first diagram, shoot tip in the media, then shoot callus you will find, and then growth of a stem you will find. So we are going to develop the stem portion, okay, and then root formation we develop. Means through changing the media contain high concentration of oxygen here in case of development of root. But in this case we are not utilizing high concentration of oxygen. What we are utilizing? High concentration of cytokine. Now Hardening means development of xylem and phloem in that developed seedling in greenhouse condition, glasshouse condition, and then whole plant developed in the atmospheric condition. So this is the development of stage of organogenesis, means development of particular organ. It can be a root or it can be a stem. Generally, we develop a stem first and then root later on. So the stem always grow only if there is a high concentration of cytokinin in the media. First, from the explant, we develop the callus, and from the callus, we develop the shoot, and from the shoot, we develop the rooting system. After changing the media, that media contain high concentration of oxygen. In this three stages, up to two, the equal amount of both oxygen and cytokine. But in this case, the high concentration of cytokine. In this case, we apply the high concentration of uh, oxygen, and then we develop the whole plant. So you already know hardening means development of roots, xylem, and phloem in the plant to become the plant autotrophs. Till now, up to stage 4 and 5, it is generally now in the stage of heterotrophs, means they are taking the nutrient from the liquid, and they not proper development of xylem and phloem. But in this stage, there is a development of xylem and phloem, and plant is able to absorb the nutrient through the rooting system and take the CO2 from the leaf and do the process of photosynthesis by in presence of light. The somatic embryogenesis, somatic development of whole plant from the explant. It can be somatic cell, it can be root part, it can be leaf portion, that is the apical portion, that is the explant. It should be uh, meristematic in nature. So we try to know. Somatic is a process by which somatic cells or tissue, including haploid cell, develop into embryo and to regenerate the plant. Means we are not going to utilize the seed, the fertilized portion, that is the embryo of the seed of the. So first induced in embryogenesis through suspension culture, it was reported by Stewart in 1958 in carrot. So that is the somatic embryogenesis. And again, the scientist Reinhardt in 1959 produced embryo from callus in carrot through suspension culture. So try to remember who had first reported the somatic embryogenesis. Stewart. Okay, and Reinhardt in 1958 and 1959. So try to know, but which plant? In the carrot. Importance of somatic embryology: high propagation rate. We cannot multiply the plant through normal embryo, means through seed. After fertilization, there is a formation of the seed. That seed used as an embryo, and we through conventional breeding method, we apply the seed in the soil, and then it will develop in the whole plant. So it will take time, and we cannot multiply rapidly, but we can multiply the same quality of plant through this somatic embryogenesis, and it is suitable in suspension culture media, artificial seed production, somatic embryo. I have already discussed artificial seed, means encapsulation of calcium alginate on the surface of somatic embryo, that will act as artificial seed. So somatic embryogenesis, that application means rapidly multiplication of same quantity of plant, same quality of plant, same number of the plant, means multiply the plant in very fast rate with the same quality through somatic cell, not reproductive cell. But in case of artificial seed production, it is utilized. The somatic embryogenesis process is going to utilize this in artificial seed. Try to know what is artificial seed. If there is a flooding occur and whole plant is going to destroy, so what is the alternative? That is the artificial seed. We can grow the plant quickly within a 5 days, 10 days, 12 days with the help of this artificial seed inside the lab, and then multiply in the uh, greenhouse, glass house, and then we give this plant for 4 months for again transplanting. And it is also labor saving, means that is required, means during the agricultural process, during conventional methods we apply the seed for multiplication of plant, but here only from one leaf we can multiply 1,000 plant, millions plant from one leaf, means each cell is going to develop into whole plants. So this is the application, this is the advantage, means rapid type of somatic embryogenesis: direct somatic embryogenesis and indirect somatic. What do you mean by direct somatic embryogenesis? Means we are going to directly produce the embryo without formation of callus directly from explant due to PDDC cells. Means you are going to form the embryo instead by passing the callus formation. Then generally we are not going to produce the callus. It will bypass the callus formation and direct form the embryo. That is known as direct embryogenesis. And indirect somatic embryogenesis, callus from the explant take place from which embryo are developed. There are two ways. First is without formation of callus, develop the direct formation of the embryo, somatic embryo, and develop the whole plant. Second, indirect somatic embryogenesis means first from the explant we develop the callus, and from the callus we develop the embryo. So that is the that is the indirect method of somatic embryogenesis. Try to differentiate what is the difference between somatic embryogenesis and that is the direct method of somatic embryogenesis or indirect method of somatic embryogenesis. So somatic embryogenesis is monocot and dicot, that there is some demarcation. We will find the such characteristic feature: radical protected by chlorhiza and plumule coleoptile and plumule. So radical, that is the root system, and plumule, that is the stem system, that is protected by chlorhiza and coleoptile. Try to know the botanical term. And cambial tissue are absent in case of monocot. So there is no secondary growth. Cambium tissue is responsible for secondary growth. So these are absent in case of monocots. Secondary growth are absent and easy to culture in the media. But in case of dicot, chlorhiza and coleoptile and chlorhiza are absent. Cambial tissue are present. So secondary growth are present. Difficult to grow in the media, means if there is secondary growth occurred, it will cause a problem for the multiplication of that plant in case of dicot. But multiplication of monocot plant is very easy through plant tissue culture from somatic cell. So try to understand. So this is the direct somatic embryogenesis, that we are going to develop the whole plant from the somatic embryo directly. But in this case, first we develop the callus, and then callus develop the somatic embryo, and then we develop the whole plant. So this is the difference in direct somatic embryogenesis and indirect somatic embryogenesis. So this is the diagram. Means there is no formation of callus here, but here there is a formation of callus, undifferentiated. We cannot demarcate it is the root or system or a stem system. तो आपको समझना है कि क्या डिफरेंस है डायरेक्ट सोमेटिक इम्यूनोजेनेसिस में इनडायरेक्ट सोमेटिक इम्यूनोजेनेसिस. ये स्टेज है, स्टेज ऑफ सोमेटिक इम्यूनोजेनेसिस. सोमेटिक सेल से सेल डिवीजन जैसे जैसे मल्टीप्लाई हो गया धीरे धीरे, ये ग्लोबुलर स्टेज बताए थे ना कि गदा शेप का बनेगा, फिर हार्ट शेप बनेगा, फिर जो है ये जो है टॉर्पेडो स्टेज बनेगा. सो दो जस्ट फॉर फॉर्मेशन ऑफ व्हाट कोटिलेडन्स, मींस cytokinin play a vital role for development of different stage of the somatic embryogenesis. So this is the different stage. I have already discussed this. Somatic embryogenesis, it is the efficient plant regeneration system under in vitro. In vitro, I have already discussed. What is the meaning of in vitro? Means in lab condition, we can rapidly multiply the same plant population with the same quality, with without infection of virus, bacteria, or fungus. It is a potential useful tool for genetic transformation. Means we can easily transform the gene through the somatic embryogenesis method. Cross-linking between hormone and transcription factor is likely to play an important role in somatic embryogenesis. Means advantage of this somatic embryogenesis is that within a very few days we can rapidly multiply the plant with the same quality, because we are going to multiply plant from somatic cell, not reproductive cell. Okay, so we can get the virus free plant if there are adverse environmental condition coming in the environment, like flood, like excess rainfall, and whole plant seedling is going to ruined, बर्बाद हो गया, तो क्या अल्टरनेटिव है? सोमेटिक इम्यूनोजेनेसिस फॉलो, यू फॉलो द सोमेटिक इम्यूनोजेनेसिस एंड वी यूज द artificial seed from the somatic embryo and multiply the plant within 14 days, 10 days you can use this, or within 5 days for getting the seedling, because seedling is the essential for multiplication, for production of cereals, for production of pulse, for production of vegetables, for production of fruits. So the mechanism of plant embryogenesis is unclear, and the comprehensive work is in future is necessary to study within the interaction of various factors for entire picture of regulatory mechanism of embryogenesis to be transparent. Means in plant system the mechanism is under a study of mechanism is very complex, so you can not easily understand how actually the mechanism is going on, but in lab condition we can do easily how it is going on inside the cell, inside the tissue, for getting the whole plant, for getting the particular organ, either a stem portion or root portion. So organogenesis and somatic, what is the difference? In part we can produce particular organ through organogenesis, we can get the whole plant through somatic immunogenesis. इसमें क्या है कि vascular system connected to parent tissue organogenesis में, लेकिन इसमें क्या है, vascular system not connected to parent tissue, समझे? Organogenesis is property of the somatic tissue. Somatic immunogenesis retained to be reproductive tissue. So organogenesis is a popular process, somatic immunogenesis is relatively less popular. Yes, we can easily produce the particular organ with the help of plant tissue culture, but somatic immunogenesis is less popular. So try to know the difference. It is unipolar structure or shoot or root primordium formation. Here we can going to produce both bipolar structure, root or shoot. Alternatively, first we produce the shoot and then we produce the root. तो ये आपको समझना है. एप्लीकेशन क्या है ऑर्गेनोजेनेसिस का, इसका इजिली आप प्रोड्यूस कर सकते हो, वेरिएशन इज नॉट प्रोड्यूस, आप वेरिएशन नहीं कर सकते हैं. यूज एज ए बायोरिएक्टर मींस यू कैन प्रोड्यूस एनी केमिकल कंपाउंड इनसाइड द लिविंग सेल, दैट इज द प्रोटोप्लास्ट कल्चर. Means cell without cell wall, you can multiply in the liquid media, suspension culture, and then protoplast culture. We can easily manipulate the gene, we can easily transfer the gene, it is helpful for gene making the transgenic growth. Okay, so this is the application of organogenesis. And advantage of organo क्या है, ये cheap है, fast है, ये आपको पता है, fast भी है, ये आपको पता है. Yeah, so try to know what is going on. No variable, because we are using the somatic cell, and somatic cell that is true in diploid condition, you are not going to use the reproductive cell, so that's why we cannot expect the variation. More variation means least amount of variation you cannot do. And less space required means space is also very requirement of space also very limited. So you can produce the large number of particular organ with the help of chemicals in presence of particular hormones. If you want to produce the root, then we use high concentration of oxygen. But if you want to produce the shoot, then you use high concentration of cytokine. But if you want to produce the callus, then equal amount of oxygen and cytokine. So try to know. Now somatic hybridization. For hybrid seed production, टाइम हो गया क्या?
Yes, sir.
हाँ, तो आपको ये समझना, नेक्स्ट डे हम सोमेटिक हाइब्रिडाइजेशन बताएंगे. सो ट्राई टू अंडरस्टैंड कि क्या क्या मेथड्स है. पहला है ऑर्गेनोजेनेसिस, दूसरा है सोमेटिक एम्ब्रियोजेनेसिस. ऑर्गेनोजेनेसिस में पार्टिकुलर ऑर्गन बनाते हैं, आइदर पहले हम स्टेम बनाते हैं, उसके बाद रूट बनाते हैं, देन होल प्लांट डेवलप होता है. particular organ ही बनाते हैं in case of organogenesis, but in somatic immunogenesis whole plant we are going to prepare. पहले हम स्टेम बना लिए, स्टेम पोर्शन, पहले तो हम एक्सप्लांट से कैलस बनाए, कैलस से क्या बनाएंगे, स्टेम या सूट, उसके बाद जब सूट डेवलप हो गया तो उससे सूट बनाएंगे. So this is the advantage कि ये quickly we can multiply the plant. आप इजिली प्लांट को मल्टीप्लाई कर सकते हो. इट विल नॉट डिपेंड अपॉन द सीजन, एंड वी कैन यूज ओनली द सोमेटिक सेल, so we cannot find any variation. We can maintain the quality of that plant from which we have started the experiment, this plant tissue culture, because in case of somatic cell there is no change in the quality, but if you are utilizing the reproductive cell there may be deterioration in the quality of the product, like cereals, seed shape, seed production, or like vegetable production, means shape, size and quality taste, it can degrade if you are utilizing the seed. But if you are utilizing the somatic cell, there is no chance of degradation of the quality of any cereals, any vegetable, any the fruit bearing plants. So this is the most effective way to get the virus free plant and rapidly multiplication of the plants through plant tissue culture methods. Okay, so try to observe what are the different methods for production regeneration of the plant. That is, one is organogenesis and second is somatic embryogenesis. तो ये जो है मैं क्या क्यों बता रहा हूँ, क्योंकि जस्ट ऑब्जेक्टिव ऑफ प्लांट टिश्यू कल्चर इज टू रैपिडली मल्टीप्लाइंग द plants for getting optimum production of cereals, vegetables, then fruits, and then the medicinal compound we can produce through this plant through molecular farming. So objective is to rapid multiply the plant or plant organs. Plant organ can be root part or stem part or leaf part, any part, through organogenesis. It was first reported by किसके द्वारा सबसे पहले दिया गया है ये सोमेटिक ऑर्गेनोजेनेसिस, स्कूग एंड मिलर. एंड सोमेटिक इम्यूनोजेनेसिस किसके द्वारा दिया गया है, किसके द्वारा दिया गया है सोमेटिक इम्यूनोजेनेसिस, बताया ना तुम लोग को, किसके द्वारा बताओ, याद नहीं है ये, you will find this is Stewart and Reinhardt. So try to remember some name of the scientist, it can be asked from CBSE. So these are the advantage of organogenesis as somatic embryogenesis. Okay, so please go through the chapter on the plant tissue culture and go through the topic, the regeneration pathway. Next day I will discuss this next topic, that somatic hybridization. Okay, means what is somatic hybridization and what is the advantage of somatic hybridization, what is hybrid, what is cybrid, fusion up to different type of variety of the cell, that is hybrid, where both nucleus will fuse and cytoplasm will fuse. But when you are going to fuse one cell that have no nucleus and other cell that have nucleus, then it forms the cybrid. So thank you very much for attending the class, and now you give your attendance within a time, because they are observing whether you are giving the attendance on the same time within the period or not. So please go through and proceed for attendance. Thank you.
Thank you, sir.